good morning to all. Namaskar. It is my great pleasure to share my few thoughts in these special occasions. For a long way, I started my journey from India. So I am presently rendering as professor in pharmacology department, and my research work is plant chemistry and as well as even the pharmacological screening of some phytoconstituents. With this related, I am talking my today's I am going to deliver on the seminar of isolations and characterizations of bioactive molecules, and it's an antineoplastic and anti-cancer activities of Justicia beddomei leaves. See, my present work has been divided into two parts. One is devoted to study of the medicinal plants, and as well as devoted to part B is devoted to antineoplastic activities of the crude extracts and isolated the molecules. See, because of the sharp growing of the populations and throughout the world, the human beings is suffering the many of the elements, so-called diseases. Because it is due to the deforestations and the microbial war has been started in the universe and resulting into hazards of the diseases which are attacking to the human beings. This is a challenging task. See, for these reasons, the people we are using many of the synthetic analogs in the terms of chemotherapeutic agents or an antibiotics or analgesics or cardiovascular drugs and etc. etc. And of course, these synthetic analogs has plays a significant role, and as well as both an activities like adverse effect means untoward effect, and as well as which induces the drug interactions. To minimize of these kind of a drug and in interactions, so we alternative medicines is only the phytoconstituents. Only the phytoconstituents. So with these phytoconstituents, we can minimize the side effect. We can minimize easiest and safe and low cost medication can be provided for underdeveloped and developed countries. And of course, after the GATT implementations in throughout the world, the many of the pharmaceutical companies come forward to embark the ambitious program to screen of phytoconstituents and for uses of chemotherapy and as in terms of even cardiovascular drugs. So with this concern, my work is connected to a phytochemistry and as well as isolations of some novel compounds. So, and of course, I have been selected as a Justicia beddomei. This is usually available in most of the all part of the Karnataka and Kerala of the India. Probably it is also available in even America also. And it belongs to this Acanthisia, and the chemical constituents is not isolated and not reported as right now. But whereas in traditionally, then in Indian Ayurvedic pandits and local pandits, the people are using for disorders of the growth and as well as to healing of the drugs as anti-inflammatory, antibacterial, anthelmintic of the compound. These leaves they are using. So this is a most the beneficial plant, and this is a picture of a Justicia beddomei. See, this plant I have systematically I have presented my lecturing with this of the phyto study as this concern of Justicia beddomei leaves. First, the plant authentication. The plant, any plant and activity will be performed unless the completion of the plant authentication we are not supposed to be carried out. With the help of taxonomies, we have plant as authentified and the specimens we have collected in my research lab. So after collection of the plant, the plant is subjected to be dried and very perfectly, not under the sunlight. Then after the collections of the plant and dried, make the powder and then subjected to a hot oxalate extraction, the process by increasing of the polarities of the solvents like a petritier, a chloroform, and absolute alcohol, and then aqueous. So these are all increasing polarities of the solvents we are used. See, for the total gram, 1 kg of the powder has been subjected for hot extractions. The yield we will got in petither extract around around 50 gram, and chloroform which is in around 40 gram, and 35 grams of ethanolic extract and 25 grams. So usually the color also you can see observed here, the number yellow color, dark color, brownish, and dark. See, usually what exactly in after the extractions is over, this is about 30 cycle, this is about 40 cycle, and this is about 40 cycles. After the extractions has been completed and dried and subjected to phytochemical evaluations, because the compounds which is usually have the properties only the secondary metabolites. So when I subjected to preliminary phytochemical evaluations in the PET ether, we obtained the compounds like stearides and saponin glycosides and fixed oils and the fats. So the chloroform, which also consisting of little quantity of alkalides, saponin glycosides and coumarin, and ethanolic extracts, a most powerful components like an alkalides and triterpenoids, and usually water extracts, carbohydrates, proteins, gums, and mucilases, which don't have any biological inactivities. So that I have selected targeted on ethanolic extract and the chloroform extract. So only these two extracts which consisting of a secondary metabolites, hence we have taken the challenge to isolations of a components, and we have subjected to for the spectral the studies as concerned. See, the isolation we has been performed by column chromatography using of a silica gel of 100 to 200 mesh, and then see what we have done. So this is a way had we performing. So 65 centimeters length of a column chromatography is filled with a silica gel, and the ratio of 9:10, 8:20, 70:10, 6:4, and 5:5, 4:6, 3:7, 2:, see like this, and 1:9. So in this profession, for in this proportion, and we have to mix with these different types of increasing polarities of the solvents, and we'll go for the elutions. So there was no yield is obtained, and only when you mix with the ethyl acetate and the methanol, we have obtained 1,000 gram and 1250 gram, and as well as 550, 350. These are the two important. So this is a negligible one, and these two will consider and to be for a fractions. When you subject it to for the TLC, thin layer chromatography, and paper layer chromatography, and even that we couldn't get any single spot. So when you get it a single spot, it is to be considered as a tailing effect. It indicates called a mixture of a compound. So that the same compound had been selected, then other column chromatography using different of the ethyl acetate and methanol we have eluted, and 400 grams of obtained a 60:40 ratio. When we have undergone for this subjected for the TLC, then we'll got the single spot and RF value which is matched with particularly like a glycosides, so that we have targeted for this isolation. When you have got a single spot, it is 100%, it is a pure compound. Similarly, this is I named as a coined out the name of GBF1, that is Justicia beddomei fraction 1, and similarly Justicia beddomei fraction 2 obtain the 200 grams. So once these two compound we have been obtained, and we have characterized the compound by various spectral studies like the melting point, the ultraviolet spectrum, and IR, that is infrared spectrum, nuclear magnetic resonance, that is proton NMR and C13 NMR, and the mass spectrum. So we have obtained the mass, particularly the UV, this is an UV, and lambda maximum of methanol, 400 we got it, the another second compound 22 or 222 we got this one. So this IR most is important one is IR, infrared spectrum, and from this we can find out different types of the functional groups, we'll get it. So these indications of hydroxylic groups. So this the characteristics of there is a presence of a hydroxylic group of 33379 and unsaturated carbon and as well as 2081 centimeters and CH stretchings will get, and carbonyl compound at 1714 will obtain. So this is a clear indications how many functional groups which are present, hydroxylic groups and carbonyl groups and CH stretchings, so unsaturated things will obtain. So by the proton NMR we will be confirming that a number of hydrogen present in the isolated the fractions. So there are 3 ionic and amoric protons in the delta value of this one, we got it, and then as well as a bunch of the signals between a delta of 350 to 390 indicating presence of a glucoside protein. So this is the most important one we have got it. This is a glycoside chain which is present, and delta 4.442, and this is unsaturated proton, and the signals of 1.8, delta value of 1.26, 1.22 and 1.06 were due to the methylene and methane protons. Similarly the signal cells 0.95, 0.8, 0.74 which is due to the presence of 3 methyl groups. So that we have confirmed a number of A protons. Then C13 proton NMR for the JBF1, it is due to we are confirming of how many carbon atoms which are the presence. So there are see that in the delta value of 170 it's due to the carbonyl carbon, probably lactone ring, and delta 126 and 142 it is due to the carbon carbon double bond, and then delta 108, 1.3, 93, 21 due to 3 anormic carbon atoms which supports an assignment the made in a proton NMR of the Justicia beddomei fraction 1. Now the last one is the confirmations of the molecular weight. Confirmations of the molecular weight. So the molecular weight is confirmed by the mass spectrum, the same isolated the compound of as a small quantity of the 5 micrograms when you subjected, and we will got the molecular point of 763. See 763 we will get the things. Yes, the pointer is not working. The 763, this is a probable compound of the JBF1. Similarly we have performed, and then the mass spectrum fragmentation also we have performed. So once the mass spectrum is performed, then only we will confirm that the compound whether is working or the not. See the 763, then we split off this compound and this molecular weight of 690, then another one we mix together all, we will get a one compound. So this is what we can call as a fragmentation. Yes, this is a one is over. Then similarly the another isolated compound of GBF, that is Justicia beddomei fraction 2, and this we have been wavelength of 230 we can found. Similarly the IR, the functional groups of hydroxylic groups we will obtain, and then unsaturated carbon group, but here isopropyl group, the additional group, the functional group will obtain. And the proton NMR and the number of the hydrogen protons is quite similar, but little difference is in the delta value of 0.75, 0.79, 0.83, 0.88 and 0.96, 1.7, 1.24 due to methyl groups of triterpenide nucleus. Triterpenide nucleus. And the brown signals of the delta 3.45 is due to the hydroxylic group of hydroxyl methyl proton of a 2 sugar units. Means the compound also is consisting of a sugar molecules. The single at the delta 4.2 and 4.27 are due to the anomic protons of the sugar units. The multiple signals of delta F41 due to hydroxyl methyl group, and similarly the unsaturated proton also found in the multiple delta 5.34. This is a C13 protamer, and we have confirmed number of carbon atoms 102 and 101.37 of the delta. There are 2 anomic carbon atoms, and delta of 108, delta 135 unsaturated carbon atoms we have been found. See delta 84.72, 76.84, 75.14, 71.74, 66.50 is corresponded to a glucose unit. Similarly, delta 81.68, 76.60, 74.41, 71.50 and 66.38 are due to the second glucose unit, and 64.53, 77.23 were due to the hydroxyl methyl carbon and hydroxyl methyl carbon atoms respectively. So the mass spectrum has been carried out, and this is we were obtained of 766.966 of the molecular weight will obtained of the compound 2 of the Justicia beddomei fraction 2. Then the same fragmentation also we have been carried out. Yes, so now we have carried out the probably the isolated molecules or triterpenoids. The second in an aspect in the second section, which is devoted to pharmacological screening. There are 2 things I have been considered. One is antioxidant activity and antineoplastic agents. So usually an antioxidants which inhibition of the oxidations of the molecules. So usually all living organisms utilizes the metabolisms, aerobic metabolisms, and for what there is a source of an energy. So this source of an energy, what happens, which resulting to the formations of the oxygen oxidative reactive derivatives. So called, which are called as a free radicals, which are so called as a free radicals, because oxygen is a highly reactive atom. Oxygen is a highly reactive atoms. So this is capable of and becoming a part of a potentially damaging of a molecules, damaging of a living molecules, so called as a free radicals. So these free radicals which are damaging of the macroles and biologically important the macromolecules, which will including DNA, which including even proteins and including lipid profiles. See, we'll obtaining the free radicals even from the nicotine, will occurring from the free radicals from the even sunlight, and see by pollutions and UV radiations and UV radiations. So what exactly these things which are all damaging of the living cells, damaging of the living cells. Now living cells means DNA damage, protein damage, and lipid damages. So the free radicals which includes, so what do you mean by free radicals? So these oxidants which are nothing but an oxidations, the chemical reactions that takes place and which transfer the electrons or the hydrogen, and which form a substance for oxidizing agent. This oxidizing agent which can be produced free radicals, which produces the free radicals. When produces the free radicals, which will able to be a damage of a healthy normal cells. So all vital and organs which consisting of all type of cells, livers which consisting of reticuloendothelial cells. So lungs which consisting of alveolar cells. So cardiovascular systems which consisting of a number of myocardial cells. So this is all cardiac automatic myocardial fibers, non-automatic myocardial fibers. So these are all what is the which leads, which are most important, the cells. When the cells are damaged, usually injury will takes place. When injury will still takes place, what happens? The liberations of a phospholipidose. So this is a phospholipidose when liberated, and is called as inflammation due to the release of the secondary autocytes like bradykinin, prostaglandin, histamine, calcitonin, so kinin and all etc. So it leads to be a ischemia so called. So when with these anoxylizes like thiols and extract the hydrogen atoms from the fatty acids, then so the free radicals usually like superoxides and hydrogen peroxides and as well as hydroxyl radicals which are responsible for the damage. So once which is a damage, what happens usually which may lead to a lot of the problems. These free radicals which are responsible for the atherosclerosis which resulted. Atherosclerosis is nothing but in the artery there is a formation of the flat flacue in the arteries. When flat flacue which has been formed, then which leads there is no good blood supply. Artery carries a oxygenated blood. Artery carries an oxygenated blood. When there is a sufficient amount of a oxygen blood will not supply to the cardiac muscle, then what is will result? Ischemia is resulted. Necrosis, cell death is resulted. Similarly, even diabetes mellitus also resulted even in the case of the pancreas. So pancreas is consisting of, do you know that there are two types of the cells, alpha cells and beta cells. Alpha cells which secretes the glucagon and beta isolate cells of the pancreas of the Langerhans will secrete the insulins. So this is damage of the beta isolate cells. What is the resulted? Insulin deficiency is resulted, diabetes mellitus is resulted. Diabetes mellitus, it is a clinical syndrome characterized by disturbance of the carbohydrates, proteins and lipid metabolism and even the deficiency of the insulin. So these all tragedies which leads the free radicals, and shock also sometimes in the olden age which is also possible in the nerve cell damages. And then a convulsions. Convulsions means it is the involuntary spasmodic contractions of the skeletal musculatures. The seizures are unobtained. So this is all tragedy with the free radicals. So that free radicals which has prevented by what a compounds is so called as antioxidant. So this concern, this is a behavior of the antioxidant. So with this concern, I have pointed out that I have been worked and antioxidant and as well as antineoplastic the properties. So screening methodology. So any preclinical studies we have carried out with as per the OECD guidelines, we have to fix the dose by acute toxicity studies. So we worked on there is usually either the sex of weighing of 25 to 30 grams of the mice was used for experimental purpose. And then as per OECD guidelines, we got 2000 mg per kg body weight and 500 and 1000. This is the dose we have been fixed for the 100. This is the lethal dose 1 gram or 1000 mg we obtained for the study as concerned. Yes, antioxidant and activity will be performed is by the different methods. That is a percentage of the scavenging we have to be carried out. So this is will be carried out by DPPH methods. DPPH means 2,2-diaphenol 1,1-picaryl hydroxyl we are using as a free radicals. So these free radicals we have prepared. We have a prepared and reduced to be corresponding the hydrogens, and when reacts with the hydrogen donor and change the yellow colors with the drug and as concerned. So how we have prepared the first the extracts and the fractions we have taken 250 mg of the next extracts dissolved in 50 ml of ethanol containing of 5 mg per concentrations of the drugs like 20 microgram, 40 microgram per ml, 80 microgram per ml, 60 microgram per ml and 200 microgram per ml of the both blank. Blank means only DPPH. Standard we have taken as an ascorbic acids, and this is all the stress drugs like ethanolic extract and the chloroform extract. Because why you have selected only these two? Because these two is consisting of a triterpenoids and flavonoids and alkaloids. So secondary metabolites. So that we have selected of these ones. So after the samples we have been prepared, and then a 3 ml of ethanolic the series of we have collected different concentrations we have taken into the volumetric flask, 10 ml of the volumetric flask, and we have taken these concentrations of the test drugs and with add with a ethanol of a DPPH will added and incubation for 30 minutes and dark light. Then afterwards we subjected undergone for UV spectra lambda maximum of 5717 nanometer. Then absorptions we will get it different and absorption. Then we have calculated the percentage of the scavenging, the percentage of the scavenging. So the percentage of the scavenging has to be calculated by A1 minus A2 divided by A1 into 100. A1 means absorption of the blank and A2 is the standard, so that we will get these things. Where is the percentage of scavenging is more, and it is a more beneficial antioxidant effect, because it is scavenging of the color has been changed. See 91% in the highest concentration of 200 will show that, and the same similarly in the 200 microgram per ml and 60, and whereas in the chloroform extract will got less. It means ethanolic extract is more beneficial, but as compared to the standard compound is not much more, but as compared to the blank, and this is very very good antioxidant and activities. So similarly we will perform the same dilutions procedures we will carried out even the fractions, because the fractions whatever we obtain different concentration like 20, 40, 80 and as well as even 160 and the 200. See after the selection of this we have to conduct it, and the same the standard and the JBF1, then so it's not functioning, JBF1 and JBF2. So this is 59.3 and the standard 91 and 57.3 and the 200 microgram per ml will got a very good the percentage of the scavenging. It indicates that very good antioxidant agent. So this is all the series of dilutions we have carried out and we'll got this. See the another one technology we have used SOD method. So this is a superoxide methods. Then what we have done in this case, the superoxidations and methods, usually this is an enzyme. This is an enzyme. It can catalyze the superoxide ion of the oxygen and hydrogen peroxides. So these enzymes which are present usually in the aerobic cells and the mitochondria, so which are enabled to be reduces the lifespans. These are the enzymes which reduces the lifespans. So become a sensitive hypoxia means difficult or a demand of an oxygen is reduces in the circulation of the blood. When an oxygen circulation is decreased, what happens? It is very difficult to be survive, or the cells will go on to be undergone for the necrosis. So what exactly in the SOD method we have not taken a drug standard easily. So whatever anyhow we have planned for the antineoplastic agent, anti-cancer drugs, so that the same animal we have selected for this. So EAC, that is Ehrlich ascitic carcinoma tumors, we are induced the same of the tumor from the animals. We have collected the blood from retroorbital vein, retroorbital vein. Then retroorbital vein of the blood when you collected, then subjected for the 6000 rpm and separation of the blood plasma. After separation of the blood plasma, the same dilutions with the serial of this one, then we have add the SOD, then the same with the UV spectroscopy, then percentage of the inhibitions we have been unabsorbed. So this is a result. JB at the 500 microgram ml and 88% and 85% of this one, and then JBF also is got a 57%, and antioxidant activity, it is due to the either triterpenoids or the flavonoids of the substitutions of the pattern of hydroxyl groups. So these are all effective free radical scavenging and 3,4 ortho-dihydro configurations in the ring 4 and 4 carbonyl groups, giving a catechol like a structures ring C, is also beneficial for a antioxidant activity of triterpenoids. Probably the aerial part of this Justicia beddomei and the fractions are very good beneficial antioxidant. This is my conclusion is very good, excellent, beneficial antioxidants can be used. Yes. The another one is pharmacological evaluations have been performed, that is known as antineoplastic inactivity. Cancer is cancer is abnormal growth of the tissues in any part of the body. But all tumor formations are not cancers. There are two types of the tumors. One is benign tumor, another one is malignancy tumors. The malignancy tumors is so called as a cancer tumors. Malignancy tumors are considered as a cancer tumors. The disorders of the growth is to be classified into a quantitative and qualitative. Hypertrophy, atrophy, hyperplasia, atroplasia, and metaplasia, dysplasia, and neoplasia. Any new thing, neoplasia means new thing formation in any part of the body is called as the formation of the tumors. Formation of the tumors. So usually tumors, how that takes place? So because of the modern civilization facing the lot of the problems, particularly with these cancers. So mutations will takes place. How? Usually it will be geometric progressions. Cell multiplication will takes place in the malignancy tumors. So it is in the geometric progressions. 2 into 2, 4, 4 into 4, 16, 16 into 16, like that. So proliferations means the multiplication of the cells very progressive the manner. You can observe how the cells are proliferated, single cell proliferated, and mutation of the DNA repair. Then oncogene is also more potentiated, and then cancer is an abnormal growth is resulted. Abnormal growth is the resulted. Of course, cancer is due to free radical, cancer is due to the chemical agent, cancer is due to the carcinogenic agent, even potentiation of the gene. There are gene which is there, oncogene, P53 gene. If it is more inactivated, then there is a chance of abnormalities of the growth takes place in any part of the body. So we have targeted and to be only simple preclinical studies and how the prevention or the regression of the growth of the tumor. So we have been collected in these ones. So usually this inactivity will be performed by we induce the tumor in the animal model. One is a control group, and the second one is at the standard group, that is a cancer induced group. Then extract 2 doses of 100 mg and 200 mg, and then the fractions also we have collected of the 2 fractions. So each group consisting of the 6 animals, all the animals we have injected except the control group. Except the control group, we have injected the EAC. EAC means early ascitic carcinoma tumor. So this carcinoma tumor of the 0.5 ml we have induced for all the animals, day 1, and we have been observed. So we have, after the induction of this one, we have to weigh the weight of the animal weekly and measure the tumor growth. So intraperitoneally we are injecting, then using of calipers we have to measure the weight of the tumors length into width squares. The volume will weekly we have to be measured and to be record, and also we have to be calculate the lifespan, how many days which will be survive the animals. So this is a treatment of the day 1. So this is a MDT is a maximum tolerance dose. This is a maximum tolerance dose is about 2 gram, about 2 gram. TI means therapeutic index, and this is a dose 100 and 200, the minimum dose and the maximum dose. The SGD means that is a drug treated group minus a drug control group. So this is what we will go on the TZD. This is the increasing lifespan percentage. See, in the day 1, if you injected for the minimum dose with the maximum dose, so we will obtain out of 6 animals, only 3 animals are survived for the 100 mg. Then after the week and even 3 weeks completed, out of 6 animals, all the animals are survived. All the animals are survived, and also increase the lifespan of 19%. You can absorb, and then SGD is also very less, so that it indicates that 200 micrograms of a plant extract is more beneficial in connection to antineoplastic properties. The similarly day 1, 5 and 9 and all will getting the same out of 6 animals which have been survived. So this is even the extract of the chloroform extract, and GBF2 we have been only the selected because GB1 we have not selected because of the amount of the compound we couldn't get completely. It is insufficient amount. Then we have subjected only JBF. It is also out 6 animals are survived, an excellent life, increase the lifespans 33.3 and then 71 for this one. The low dose we have selected 30 mg, 60 mg, 30 mg and 60 mg. So it indicates the relatively these bioactive molecules, particularly triterpenoids, has got an emerged with approved and effective anti-proliferations and effect against the early ascitic carcinoma tumors in the mice. Both the extract of ethanolic extract and chloroform of JB contain of the triterpenoids as more responsible bioactive molecules which inhibitions of the cancer proliferations. So this many kinds of cultured human cancers proliferations we have been tried and is responsible for exhibit potent anti-tumor activity against the EAC induced mice. And the work is also we continued for the MTT bioassay and the total conclusion, it is both the compounds which has got antioxidant and antineoplastic the properties. So this a compound I have even the work is going on for MTT bioassays and the molecular level of particularly in the life cycle of a G1 phase and S2 phase DNA replication, how it will inhibit. With this concern my work is going on. So thank you for your kind listening and kind cooperations. In these special occasions, I acknowledge my special gratitude to all OMC organizers and respected research scholars and delegates, mm, and all my lovely research scholars. Thank you once and all.